So, as Williams mentioned, I'm Yazdi Pitavala, and like you said, that's a mouthful. Should you find yourself falling asleep during the next hour, repeat my name in your head a couple of times. We'll keep you alert. But I'm a clinical pharmacologist down the road at Pfizer. I got my PhD in clinical pharmacokinetics from the University of Minnesota. And following that, I did a postdoctoral fellowship at UNC and at GlaxoSmithKline at that time. And after that, I started working in the pharmaceutical industry. I started at Little Aguron Pharmaceuticals, which used to be down the road here. And through a series of mergers and acquisitions, now I'm part of Pfizer. I've been there for about 15 years and have been mostly working in the clinical development of oncology drugs, drugs for cancer. And I'm absolutely delighted to be here to talk about phase 1 studies. It's something that we do a lot and we spend a lot of our time doing. So I'm really glad to be here. Just a few things I want to mention. One is, since this is my first time here, I really want this to be very interactive. So if you guys have any questions, please feel free to throw them to me. If I'm the only one who's spoken till the end of the hour, then I'll have done horribly. So I really want this to be a discussion. Second thing is I tend to speak fast and I have an accent. So again, if something doesn't make sense, please stop me once, twice, as often as you want. You'll find it's very difficult to offend me. You'll have to try really hard. So all questions are good. Just throw them my way. Let's go ahead and get started. So phase 1 clinical studies, that's what we're here to talk about. Phase 1 means different things to different people, and that's one of the things I wanted to mention during the next hour. So here's a brief overview of what we're going to cover, and depending on how much time we have, we can delve into more detail on any of these topics. And incidentally, if any of you have questions afterwards, feel free to reach out to me as well. So I wanted to go over the terminology, speak about what the typical objectives might be for a phase 1 study, and then spend the meat of the time during the hour talking about study designs. How would you do a good phase 1 study? On the face of it, it seems like a pretty obvious thing. It seems like a fairly straightforward thing to do, but the devil's in the details, and we can go through study design issues. And then at the end of the hour, if we have time, we can delve into dose selection. How do you pick a dose for a phase 1 study? And that by itself, you could do a thesis on it that, you could spend several hours talking about dose selection, but we'll see how far we can get. So here's the crux of phase 1 studies. A phase 1 study literally is, in the most general sense, the very first time you're going to dose a human, okay? And the reason why phase 1 studies are challenging, I think, is because they sit at the crux of two objectives which are kind of competing with each other. The very first objective is, this is the first time you're doing a study in a human being. You want to get as much information out of it as possible, right? You want to find out what this drug does to humans. And so greedily, as a scientist, you want to get as much information out of it as possible. But the bigger imperative, and this is where the conflict comes, is you should never do it at the cost of the safety of the subjects that are enrolling in that phase 1 study. So the number one primary imperative of a phase 1 study is to find out how safe that drug is. And you should never, ever want to compromise the safety of those individuals. So the challenge lies in finding that sweet spot where you get as much as you can out of that study while still ensuring, very solemnly the safety of the people who are going to participate in the study. This is something which should be very, very familiar to you. This is like a general clinical drug development paradigm. And you guys should all know that you start with preclinical testing. Typically, you would do a fairly wide battery of studies. You would do in vitro studies. You would do studies in animals. You would do toxicology studies. In the old days, before you could do electronic submissions, you would literally have like 2 big trucks full of paper, which would get submitted to the FDA for an IND. So all the studies that you've done, even before you dose a human being for the first time, you would put all that information together, send it out to the FDA, and essentially at the IND stage, the Investigational New Drug Application stage, you're asking the FDA if it's okay to dose a human being, if you've collected enough information that it's feasible to move into humans. Yeah? Just from a regulatory perspective, what the IND actually grants you is the license to ship a drug for investigational use, but we all know it's actually for use within humans. Okay? Once you move into your phase 1 study, your primary imperative of a phase 1 study is to find out the safety of the compound. It's that imperative that first do no harm. So very often for the majority of drugs, even before you will ever find out if the drug has its intended effect, you first need to find out that it's safe. So before you ever say if I'm developing a drug for asthma, long before I'll ever give it to a patient who has asthma to find out if the drug actually has any effect, I will have had to find out if the drug is safe first. So you need to find out safety. And that's really the purpose of your phase 1 study. Typically small study, about 20 to 100 subjects at most. And typically there are 2 studies done in phase 1, and we'll go through that in a bit of detail. So typically your goal of your phase 1 study is to find out safety and to find out what dose you might want to take for subsequent clinical development. Now, there are exceptions to everything. 9 out of 10 times your phase 1 studies will be done in healthy volunteers. And why is that? You don't want any confounding factors? Absolutely. Absolutely. So if you do it in patients might be taking a lot of other drugs. You're concerned about drug-drug interactions. It's perceivable that a patient who has a disease already has their health compromised. And if you have an untested new drug, you don't know what the safety of that is. The perception is that a healthy person might be able to tolerate that a little bit better than a person who is diseased. So typically for those reasons, you would go to a healthy volunteer population. There are exceptions. Oncology drugs, very often you go straight into cancer patients for obvious reasons. And if you have time, we'll go through that. Then you move into your phase 2 study. And Dr. Parivar, who sits right next to me, incidentally, at Pfizer, he will be going over phase 2 studies next week with you. The goal of the phase 2 studies, this is now the first time you're actually going to test your drug in the intended population. Okay? And you want to try to get early signs whether your drug is doing what it is meant to do at the dose that you have picked. Okay? If it's a drug for diabetes, is it really reducing blood sugar? If it's a drug for asthma, is it causing a reduction in your FEV? So that's, you do a small study and you confirm whether your drug actually has the intended effect. And once you know that your drug has the intended effect at doses where you can manage it fairly safely, then you go to a phase 3 setting, which is meant to replicate the larger real world setting. And you would do a confirmatory larger set of studies in thousands of patients at that point to confirm your findings from phase 2. And once you have all of that, Godspeed, you go ahead and submit to the FDA again, even more data, and then your drug will hopefully get approved at that point. So let's jump into phase 1. Phase 1 means different things to different people. The first definition, which is based on chronology, is the most commonly used definition. That's the definition that we are going to be using for the purpose of this class. Phase 1 literally means the very first phase, the very first set of clinical studies you do. So that's typically what people mean when they say phase 1. Very often, even in the literature right now, you will find people using the term phase 1 to actually define something based not on when the study is done, but the population in which the study is done. So suppose I'm already into phase 2 or phase 3 trials for a compound for asthma, right? And while I'm doing that, I need to do a study to evaluate the effect of food on my compound. Should I take the drug with food or without food? Well, I go to a healthy volunteer population and I do a study at that point. So even though I'm during the clinical development continuum, I might be in phase 2 or phase 3, because I'm going back to the healthy volunteer population, some people might call that study a phase 1 study. Does that make sense? So by virtue of the population, anything done in healthy volunteers is sometimes called a phase 1. And then the third context in which phase 1 is often used is in terms of the intent of the study. So a great example, suppose I've developed a drug for cancer. Phase 1, phase 2, phase 3, went to the FDA, got approval, it's out in the market, everybody's using it. Now I decide I want to try it for another tumor type, okay, another indication, but this time I want to use it in combination with chemotherapy. Okay, so now I'm going with the new combination, an untested combination for the first time, and that, because you're assessing the safety of this combination for the very first time, people use a phase 1 terminology for that study as well. Okay, makes sense? But again, for the purpose of this study, we're going to stick with the very early studies which are conducted.